In this video, we will be talking about our work on precision medicine in cancer. The general problem has a simple solution if you have the right kind of data. For example, you could have omics and clinical data on a set of patients and train a model for the drug responses on those omics, and then use that model to predict the responses for the future patients. Here, we have only used proteomics. The problem becomes complicated because one can't elicit responses at will from patients by subjecting them to drugs. We suggest a workaround by utilizing cell line model systems. There are databases which offer a large variety of data in this context. We used TCPA for patient proteomics, MCLP for cell line proteomics, and GDSC for cell lines drug responses. Utilizing patient heterogeneity, we want to bridge anti-cancer pharmacological data to large-scale omics, taking into account the interactions between genes within pathways and the variabilities across cancer types and data sources, that is, the patients and cell lines. We frame this as a missing data problem since the patient drug responses are not typically available. Then, we propose a two-step solution using a multi-scale Bayesian network model called TransPRECISE. TransPRECISE first identifies matching cell lines for patients based on proteomic similarity, and then we train drug response models using cell lines only. Finally, we combine these two steps to predict patient drug responses using pathway activity summaries as predictors. This is our overall framework, and then this is our published paper where you can find the modeling details. I would now like to take you through our interactive R-Shiny dashboard for our results combining MCLP cell lines and TCGA tumor samples. The first tab provides a broad overview, instructions, and relevant references for the tool. The second tab allows you to interact with the fitted pathway networks across patient and cell line cancers. You can look at the results for 31 different patient cancers, 16 different cell line cancers, and 12 different pathways. In the third tab, you can look at the within pathway interactions across all patient and cell line cancers, again across the 12 different pathways. Here, we summarize the matching of patients with cell lines using three different approaches. You can look at their network similarity, or the correlation of network activity scores, or a hierarchical clustering based on these scores. For the drug response model, we summarize the predictive affinity of pathways across cancer sites. We also report the top responsive drugs across different cell line cancers and patient cancers. Finally, all our codes and data are available for download so that you can reproduce our results or apply our method to your own data. Thanks for your attention.